All right, thank you guys all for coming. We're going to talk about the treatment and factors that affect kids with cancer, and I hope it's going to be a really good session. The first speaker, actually, we're going to have 4 talks. 3 of them I know are going to be outstanding. One of them may be just okay. That's mine. First speaker, I am seriously delighted to be introducing to you Naveen Pinto. Dr. Pinto trained with me at the University of Chicago. We did our residency and fellowship together. And as anyone who knows me will not find hard to believe, Naveen was the good fellow. I definitely was not. and Naveen saved me a lot during fellowship. There was a time when I was actually going to leave the wonderful world of pediatric oncology, and Naveen sat me down and said, "Bob, you're the yin and my yang. You can't go." And most of the time, I'm thankful about that. Not always. But Naveen spent a couple of years at Chicago after we finished fellowship, and then went to Seattle Children's, where he dealt with solid tumors, particularly neuroblastoma. And he recently, as in 6 months ago, went to Denver Children's. He is a leader now in neuroblastoma and actually will be one of the co-PIs on an upcoming trial for high-risk neuroblastoma for the COG. Naveen Pinto, come on up.
Great. Okay. Hi, everyone. I'm Naveen. Bob, thanks for having me. And I'll promise not to tell too many stories from fellowship. So my talk is discussing how to get drugs approved in rare disease and lessons from neuroblastoma. Neuroblastoma is one of the few pediatric malignancies that has drugs approved for it specifically. And so I wanted to go through the 3 FDA-approved drugs that we have in the clinic now for neuroblastoma and sort of how they got to where they're at, because I think there are some lessons to be learned about how the different companies that develop these drugs, different strategies they took to get the drugs developed. And I think that there may be some lessons for other rare diseases. I have a couple of disclosures. I have paid advisory roles for Novartis and Eureka Therapeutics on their data safety monitoring committees. And then I listed these other 2 because they pertain to what we're talking about. I have advised both YMABs and US World Meds, but I have not received any payment for that advice. So just for those of you that aren't aware, neuroblastoma is the most common extracranial or non-central nervous system solid tumor. And we divide neuroblastoma into risk groups by both clinical and biologic factors. And what sort of fascinated me when Bob and I were in training about neuroblastoma is this really wide array of heterogeneity. There are some kids with neuroblastoma that are observed and their tumors spontaneously regress. And what I spend most of my time thinking and worrying about are the kids that are slightly older, 18 months of age or older with metastatic disease that really need a kitchen sink approach to get basically everything we know thrown at them, and still only about 50% of them are long-term survivors. So again, just to highlight how rare this is, we have only about 400 new cases of high-risk neuroblastoma in the United States per year. Our long-term survival for those patients is about 50%. And despite that, we have 3 FDA-approved therapies for this disease in particular. Those therapies are dinutuximab, naxitamab, and eflornithine. So we'll go through the history of how those drugs were developed and approved, and then hopefully give some advice about what that means for the field at large. So high-risk neuroblastoma therapy is really divided into 3 major phases. Induction with the goal of inducing as close to a clinical remission as possible. Patients that have a good response to induction therapy and are in close to a clinical remission often proceed on to consolidation therapy. And in most of North America, that is 2 very intensive rounds of chemotherapy that are myeloablative, followed by the patient's own stem cells used to rescue them. So this incredibly toxic therapy is supported with the patient's own stem cells. Patients get radiation therapy and post-consolidation immunotherapy using the patient's own immune system to sort of sop up any residual disease. And the goal of that is, I'm not sure if I can point here, is obviously to get patients as deep of a remission as possible. And like I said, about 50% of those patients will stay in remission. So immunotherapy is going to be 2/3 of the discussion. And so just a sort of primer about how we think this works. So on tumor cells, there is a target, these little green bars called GD2. That's a ganglioside that's on these developing neuroblasts that form tumors. And unfortunately, there is some of this same GD2 protein, as you can see, both on sensory nerves and pain recepting nerves, nociceptive nerves. And that's where the bulk of side effects from these GD2-based immunotherapies come from. The antibody binds to GD2 on the tumor, but unfortunately also binds to GD2 on nerves. And I put a sort of infographic of a patient using a patient-controlled analgesia or PCA button because we often have to give relatively high doses of pain meds to make this drug tolerable. So it's a unique drug, and one of the few drugs that we give that actually causes acute pain while we're giving it. So I just wanted to, again, highlight how we've gotten here. And dinutuximab was the first immunotherapy that got an FDA approval in neuroblastoma. And the trials for dinutuximab started way back in the 1990s. So the first published phase 1 study of this antibody was in 1995. And there were several follow-up studies, both in Europe and in cooperative groups in the United States. This antibody was initially called CH1418, and it is a chimeric antibody. It was developed by actually inoculating mice with neuroblastoma, and those mice, their immune system rejected the tumor. And so they were able to find the anti-neuroblastoma antibodies that the mouse was rejecting these human tumors with and develop that into a drug. It's chimeric. The sort of blue lower half on the chimeric diagram there is human antibody fragments, and so that makes it a little less reactive in patients and less likely to be rejected by a patient with neuroblastoma. So what led to the eventual approval of this drug was a very, very traditional randomized phase 3 clinical trial where patients were randomized 1-to-1 to receive either the standard of care, which at this time was after you'd been through all the intensive therapy, a 6-month program of retinoic acid or Accutane. That medicine, which is mostly used for teenagers with bad acne, also has some anti-neuroblastoma effects. That was the current sort of maintenance regimen in the standard of care in the late 90s, early 2000s. And half of the patients got that drug plus dinutuximab, plus the antibody. And so you can see how the patients were split equally. And as we discussed, you can see here, this is sort of the breakdown of side effects based on what arm you were on. And so on the left were the patients that got the immunotherapy arm. And you can just see the numbers of side effects was vastly higher, mostly pain, and low blood pressure in patients getting this anti-GD2 antibody compared to patients just getting Accutane. But this data was very compelling. I'll go back and just show you that in the red bar is the survival for patients who got the anti-GD2 antibody, 66%, and the patients that got just the retinoic acid had a 46% survival. So this was clearly an active drug and improved outcomes and led to it being considered the standard of care in high-risk neuroblastoma. So here's the timeline for how long that took. And again, very, very traditional drug development of a drug coming out of a lab. The first reports in the lab were in 1987 of this drug and how it could target neuroblastoma. We talked about the first phase 1 study being in 1995. This randomized clinical trial that was a phase 3 study opened in 2001 and took about 10 years to accrue. And it was finally published in 2010. There was some follow-up data needed to support its FDA approval that the FDA required. And finally, in 2015, we got an FDA approval. So really a 30-year timeline from first reports of the drug to it being FDA approved. And so that's a traditional sort of timeline for a rare disease. So let's talk about some of the other drugs. Naxitamab is another anti-GD2 antibody. It's a very similar drug with a similar mechanism of action. Again, just a diagram to show the differences. So we talked about the mouse antibody, of how this was first developed, of giving a mouse human neuroblastoma and watching it be rejected. Dinutuximab being a chimeric antibody that has some human fragments. And naxitamab and other antibodies are humanized. So 90% of the antibody structure is derived from human, and only small fragments that help the antibody recognize neuroblastoma are derived from a mouse. These antibodies, naxitamab and its predecessors, were developed by Dr. Chung at Memorial Sloan Kettering, and they started with a fully mouse antibody called 3F8. And they saw similar side effects to dinutuximab, but one of the unique side effects they saw with that was a rejection phenomenon, something called HAMA, human anti-mouse antibodies, where the patients were rejecting this antibody and clearing it very quickly. And so they developed this humanized antibody to sort of circumvent that problem. Now, we'll talk a little bit about this in the end, but this trial was run almost exclusively at Memorial Sloan Kettering. So unlike the dinutuximab story, where this was a national phase 3 randomized study done across North America, Australia, and New Zealand, naxitamab was really only available at Memorial Sloan Kettering for the vast majority of this approval timeline. And so the data is much more sparse because it was a single center study. This is just broad level highlights showing that some of the initial publications of this drug first being used in the clinic only had 57 patients. Several of those patients were in remission after getting some preceding therapy. And so only 26 of the patients 31 of the patients had a valuable disease where you could watch whether the drug was actually shrinking tumors or make them go away. And so the main study that led to the FDA approval of naxitamab was a study called 201. It had 36 patients in the efficacy population, and they received a median of 7 cycles, up to 11. The overall response rate, which was defined as a complete response or a partial response, was 58%, and 44% of those patients had a complete response. And of the 9 responders who had a partial response initially, some of those went on to actually then have a complete response. So I wanted to highlight this here because one of the unique aspects of neuroblastoma is that neuroblastoma can exist in a very low, sort of minimal residual disease state in the bone marrow. And if you're clearing the bone marrow space or making scans improved, oftentimes you might not have that adult RECIST criteria where you're watching a big, bulky mass shrink away. And so we've had to develop some unique response criteria in neuroblastoma of, are we clearing the bone marrow compartment, or are nuclear medicine scans getting better? And that is often our metric of success. So, and then I think the last thing I'll highlight here is that, again, just like dinutuximab, the most common adverse event was pain. And low blood pressure also occurred. You can see essentially 100% of patients that got this drug had pain and low blood pressure. So that was the initial publication that had a mix of patients with bulky tumors, some patients with remission, and they had these very encouraging response rates. It's hard to tease out from the publications, but I think most of the patients that responded in that CR group had very minimal amounts of disease, that the patients with big bulky tumors didn't really respond. And so that led to a follow-up study in Spain where patients in remission who were driven into remission by chemotherapy or something else were then treated in a remission state with naxitamab and watched to see if their tumors relapsed. And as you can see from the survival curves, the results were quite good. On the top, the event-free and overall survival for patients treated in remission was excellent. When you broke it down by remission state, this probably makes sense, but patients in first remission in the lower half on the black bar did much better than patients who were in a second or subsequent remission. And so I think how the company that developed naxitamab sort of threaded the needle to get this drug approved is they really honed in on the group that seemed to respond best. So their language around their approval is very, very specific. So I just highlighted it here. So naxitamab is approved in combination with granulocyte macrophage colony stimulating factor, a cytokine that is thought to support these GD2 antibodies' effect. For patients that are at least a year of age with relapsed or refractory high-risk neuroblastoma that is only in the bone or bone marrow, so a big bulky tumor in the abdomen is not technically approved for naxitamab, and that the patients have to have demonstrated a partial response, some sort of response to prior therapy. So they have to be responding, they have to have disease in these limited compartments in order to fit the criteria. And so that was really where this drug worked. And as you can see, by limiting the language of who was approved for naxitamab, the timeline is accelerated. So although the mouse 3F8 antibody first did appear in the 1980s, like dinutuximab, this humanized antibody first reports were in 2012. And fast forward to 2021, when these publications came out. Actually, the drug was approved in 2020, and the publications supporting the approval actually came out after the drug was approved, which is unusual. So you can see here that compared to the 30-year timeline for dinutuximab, we have about an 8 to 10-year timeline for this drug. All right, finally, I wanted to talk about DFMO, which has a completely different mechanism of action. And again, I've had to piece together this story a little bit, so I might not get all of the details completely right. But interestingly, I think where this started from is a case report. In 2006, a patient with chemo-refractory neuroblastoma who was actually on hospice developed Chagas disease, which is a parasitic infection that's common in parts of Texas and South America, and is treated with a drug called nifurtimox. And the patient got nifurtimox, and actually their Chagas disease got better, their infection got better, but they went into remission with their chemo-refractory neuroblastoma. And so the doc that treated this patient then took nifurtimox to the lab and was able to show that nifurtimox was able to kill neuroblastoma cells in culture. And when tested against another healthy cell line derived from lung endothelial cells, there was really no effect in the black bars down here of nifurtimox on healthy cells, but sort of a dose-dependent effect of nifurtimox on neuroblastoma. So how did we get to DFMO? Nifurtimox is used to treat Chagas disease, as I mentioned, and a similar disease that affects the African subcontinent, sub-Saharan Africa, is African sleeping sickness, and DFMO is used to treat that. So my understanding is that because nifurtimox worked, DFMO was also tested against neuroblastoma and had some similar activity and moved forward, mostly because it was much more tolerable than nifurtimox in terms of side effects. Since that drug has entered the clinic, I think the understanding of how it actually works against neuroblastoma actually followed the actual introduction into clinical trials. And it's thought that DFMO inhibits an enzyme called ornithine decarboxylase, or ODC1. And this seems to be upregulated in certain subtypes of neuroblastoma. It also is thought to inhibit some of the immune system that actually is co-opted by the tumor and has actually a negative effect on the patient and allows the tumor to grow, this what we call tumor microenvironment. And so we think the DFMO drug works both on the tumor itself and these co-opted immune cells that are sitting next to the tumor. So a very traditional phase 1 clinical trial was initiated for DFMO for patients with relapsed or refractory disease. Patients very standardly had to have measurable or evaluable disease. 21 patients enrolled on this phase 1 study, and 18 were evaluable for response. I apologize, it's not projecting very well, but essentially the takeaway is that nobody responded to the drug when they had measurable or evaluable disease. And the Kaplan-Meier curve on the right-hand side is sort of what you would expect for a relapsed neuroblastoma population, that there were some patients that had stable disease and they survived for a long time, but most patients did not. There was one partial response that's buried in this table. So one patient did have some shrinkage of their tumor. And so I think combined with the nifurtimox story where they saw a patient go into remission with this class of drug and this one patient that had a partial response, I think the investigators had this hunch that the drug was active, but that they wanted to treat patients in a minimal residual disease state. And so that led to a study called 03B by the Neuroblastoma Medulloblastoma Treatment Research Consortium. And this was a phase 2 study. So again, one of the unusual things here is that this phase 2 study was done as a single-arm study. Everybody that enrolled on this study, it was initially at a single site, had access to the drug. I think that was because the investigators thought the drug was active and they did not want to randomize patients to receive a placebo. So these were taking patients in remission. You couldn't find neuroblastoma anywhere when you did a bone marrow or a scan, and everyone got DFMO. So that, I think, as a parent, would be what I would want from my child. But as a clinical researcher, it is very difficult to run a study like this and interpret the results because you're taking kids with no measurable disease and saying, is the drug active? If you're taking patients with a big tumor and you watch the tumor shrink, you know the drug is active. But if they're in remission, how do you know that the drug had any impact? And so how they studied this is they did that single-arm study, and then they went back and looked at historical controls. And so how they did this is they actually used the dinutuximab data in the Children's Oncology group and looked at matched controls. So they matched kids by their age, their sites of disease, all these various factors that we know are prognostic in neuroblastoma, and took 270 kids that were matched in these categories that did not receive DFMO and 90 children that were on this single-arm study. And they compared the outcomes of the two groups. And they just looked at the data, raw data, and saw, okay, the DFMO kids on this single-arm study did better than historical controls. And then went the extra step of really this, what's called propensity score matching, where they matched patients by all of these clinical factors that are prognostic and were able to show that DFMO was effective. So you're seeing here in these patients that are in remission, 80 to 90% of these patients are not having further relapses and are surviving. And so this propensity matched analysis was used towards the FDA approval. So this is the timeline for fluornithine. The first reports of DFMO use in neuroblastoma were in 2004 after the Chagas disease case series, but people had been testing this line of drugs for 20 years prior to that. But I think this timeline should probably start around 2004. The phase 1 study opened in 2015, and the phase 2 single-arm study opened soon after in 2018. And really, from that phase 2 study to 2023 is when DFMO was approved. And again, the publication followed at the actual approval. So what are some takeaways from these experiences? I think this is the traditional timeline of how drugs get out of the lab and are approved. And I think what we saw with both naxitamab and DFMO is, if this is like a game of chutes and ladders, is that there was sort of a chute here from phase 2 clinical trials to declaring these drugs as standard of care FDA-approved therapies. So I think for people that don't think about neuroblastoma a lot, these are the takeaway lessons that I came away with, that in rare diseases, traditional phase 1 to phase 3 timelines can take 20 or 30 years or longer, like dinutuximab did. If you apply really robust knowledge of where your drug works and try to select a subset of patients that you predict will respond to this therapy, like patients with minimal residual disease, you might be able to accelerate that, like naxitamab did in 9 years, and then the use of robust historical controls can also help accelerate this process for single-arm studies. I do want to point out one caveat for at least the second and third examples here, is that in neuroblastoma, poverty is an independent predictor of poor event-free and overall survival. So poorer patients do worse. Naxitamab and DFMO were only available for the majority of their timeline at single centers. And so families had to travel oftentimes to get access to these drugs. And by nature, more well-to-do families are able to travel. And so one of the biggest sort of criticisms of these single-arm studies is that the sort of breakdown, you can see this is the actual event-free and overall survival curves by poverty, not by drug exposure. And they look like many of the curves I showed you. So the blue lines here are the poorer patients that are exposed to poverty, and the red line are those that are more well-to-do. And it's the same sort of delta between those arms as these FDA-approved drugs. And so it may truly be that we're selecting a biologically more robust cohort of patients that are going and traveling to centers to get this drug. So that's one of the bigger arguments, is these drugs might not be as active as we think. We're just giving them to a group of patients who's more receptive to them. And I would say that many patients or people in the neuroblastoma community, myself included, are skeptical of naxitamab and DFMO data for this reason. And that despite the long timeline, randomized clinical trials do really guard against this skepticism. So I think that's all I have. I included one sort of flashback slide of myself and Chicago, and I really did want to thank Bob for inviting me here. It's been a great conference, and I think I have a couple of minutes for a question, or are we doing that at the end? Okay, great. Yeah, Bob.
Can you talk just a little bit about, after brentuximab was actually FDA approved, there was still a big lag before we could actually get it in the clinic. And can you talk a little bit about that? I think people need to know that these are some of the only drugs ever FDA approved for pediatric cancer indications, right? Literally some of the only drugs that are, I can't think of any others, right? Most of the other ones were actually using off-label or were never tried in kids or for historical reasons. These are some of the few. Brentuximab showed a lot of efficacy in that trial.
Yeah, I think that it was a frustrating experience that we had this big 20% delta in event-free survival, and because of the complex nature of neuroblastoma response, of needing to look at the bone marrow and needing to look at scans, the FDA really wanted all of that source data. They wanted an independent review of bone marrows, they wanted independent review of scans, and they wanted some independent reviews of the sort of pain scale. And so there was actually, after that landmark study was published, there was a 3 or 5 year lag where essentially a phase 4 study was needed to be run in neuroblastoma to sort of prove to the FDA that you were seeing responses on scans and that bone marrow was clear, and those independent reviews were not built into the initial phase 3 design. So essentially a phase 4 follow-up. And so that delayed the drug getting into the clinic. It's still not available in most of the world. It is FDA approved, but it is not approved in Europe. It is not approved in Asia. And so many families, again, these more well-to-do families, are traveling to get access to this drug. Yeah.
2 questions.
Sure.
One, from a patient standpoint, how do we quantify the unmet medical need for neuroblastoma? And the second one, I saw this phase 3 studies done for pediatric cancer, which has 400 cases a year. What is the current trend in terms of development for neuroblastoma?
Yeah, so I think there's still I'll answer the second question first, a big push to do randomized phase 3 studies. We think that these DFMO approvals should allow us to try and push the envelope, but I would say that although the FDA has been receptive, many of these trials are sponsored by the National Cancer Institute and CTEP, and they have a very, very different view. So because they are sponsoring these trials through the Children's Oncology Group, they are really, really pushing for traditional randomized phase 3 clinical trials against the standard of care.
But are the phase ones done in adult population or pediatrics?
So a lot of times the phase 1 studies are, if we think that we have a drug, these GD2 antibodies were done almost exclusively in pediatrics, in neuroblastoma. So there is a very good track record of getting sort of first-in-human, first-in-child drugs into patients with neuroblastoma, despite the rarity. Again, if you have some idea that it will work in melanoma or something like this, that's fine. But we also, I think, have a robust network of getting phase 1 studies done in neuroblastoma, despite the rarity of the disease. And that's to sort of answer your first question. Still, despite some of this encouraging data, about half of patients relapse, and the unmet need is that at least 200 kids in the United States and many more across the world have relapsed, are not going to respond to any of the drugs I just mentioned, and so that's the unmet need. And many of those families are highly motivated to enroll in a trial. Yeah. Great. Thank you all.